Hi, my name is Yeala Shaked from the Hebrew University, and today I would like to present a long-term, fruitful and very enjoyable collaboration between myself, Benjamin Twining, Alexandra Tagdibao, and Maria Maldonado. Our paper is soon to be published in Global Biogeochemical Cycles. The topic of today's talk is bioavailability, which is in essence the link between iron in seawater and phytoplankton productivity, or more specifically, the link between iron speciation and its uptake by phytoplankton. Today I'll present a new concept to describe, probe, and compare bioavailability using a single kinetic constant, Kn applied. The concept is rather simple. Under subsaturating conditions for the uptake system of a phytoplankton cell, uptake rate is linearly proportional to concentration of iron. So in essence, in order to extract the uptake rate constant or Kn applied, all we need to do is to divide the uptake rate by iron concentrations. We have been developing this approach for quite some time now. We started with a study of Liz et al., where we compiled a very large database of published uptake rates of defined iron complexes by many different phytoplankton species. And we basically observed that all cells are reporting bioavailability in a similar way when they are normalized to surface area. We later extended this approach to a study of natural samples from seawater, again using iron-limited cultures. And again, we found that all different phytoplankton species similarly report the availability of iron in natural seawater. In the current study, we wanted to take our approach a step further and evaluate uptake rate in the ocean. Under steady-state conditions, uptake rate equals the cell iron quota times its growth rates. Over the last 20 years, Ben Twining has been sailing the ocean and collecting phytoplankton and analyzing single-cell iron quota using synchrotrons. Like many of us, we are benefiting from the geotraces revolution, and we have a very large database of iron concentrations. And finally, growth rate. We have been debating a lot how to use them, how to obtain them, and eventually decided to go with data from the Pisces model from the same location and time in which the samples were collected. Some of the important choices of the study was to choose the type of iron that we use to probe bioavailability. We decided to choose dissolved iron over Fe prime. This is so despite the fact that Fe prime is indeed the most available species. However, it is very transient and it's very hard to measure. In contrast, dissolved iron includes clearly Fe prime and a bunch of other molecules. Some are less available and some are not available at all. Therefore, it is basically a practical solution given the many unknowns in our system. To convince you that our choice of dissolved iron over Fe prime is not just for convenience, I would like to show you the current exercise. What you see here is a plot of iron quota measured by phytoplankton in the ocean in color, and phytoplankton, Fe limited of course, in laboratory cultures by Swinda enhancement. Both of them are plotted versus calculated Fe prime concentrations, and you clearly see a big mismatch between these data sets. However, when we replot the quotas of natural phytoplankton versus dissolved iron on the x-axis and leave the cultured versus Fe prime, we see a very good match. And from this data, we conclude that phytoplankton in the ocean are indeed able to sense, to detect, to access dissolved iron. The data we analyzed for this study include 8 data sets collected in 11 cruises between 2002 and 2016. The different cruises are titled here, and the specific stations are plotted on a map of iron concentration in the ocean from the Pisces model. Since K-in applies only for iron limited stations. We had to screen all the stations and distinguish which of them are iron limited, even to some degree, versus stations that are limited by nitrogen or are not limited at all. We use the data reported for each cruise. In many of these cruises, there were incubations and so forth. And also a more consistent approach of using Tom Browning's index of nitrogen over iron, where all the stations that are plotted in red are basically iron limited, while stations plotted in white or blue are nitrogen limited and therefore were not included in the analysis. For K-in to serve as a bioavailability proxy, it had to meet various criteria. The first we already discussed, that cells are iron limited or iron stress. In addition, K-in has to be proportional to cell surface area and comparable across taxonomic groups. I demonstrate how we tested it, showing data from FE cycle 2, but it applies for all other cruises as well. Basically, I plot here the K-in of every single cell analyzed in this cruise versus its own cell surface area. And you see that indeed there is a good linear relationship between K-in values and the cell surface areas. In addition, the different taxonomic groups represented in this cruise, flagellates, diatoms, and pico-eukaryotes, all fall along the same relationship, and therefore the two other criteria are met. Now for the last criteria, which is rather important. In order for K-in to serve as a good proxy of bioavailability, it has to remain unchanged at varying degree of iron limitation or at varying low concentration of iron. To test this we basically plotted the average K-in of all the cells in a specific station, and we normalized it to surface area so we can compare all of them. And you can see here in black stations that are considered to be extremely iron limited, and in gray stations that are moderately iron limited, and they all seem to agree. Therefore, indeed, our proxy is valid regardless of iron concentration or the degree of iron limitation. What you can also see in this graph is a different group of stations, of cells, that are primarily nitrogen limited. And these cells have a lower bioavailability proxy, basically since they are not upregulating, the cells are not upregulating their uptake system, and you can clearly see that they fall short of the common K-in of iron-limited cells. And as I said before, these cells were not included in the final analysis of K-in. So now, having verified that K-in is a good proxy of bioavailability, we can now look at the data we obtained. I plot here a single cell K-in for the 560 cells in the different cruises that were included in our analysis. We plot them against cell surface area. And you can see that all values plot within the boundaries of the bioavailability envelope, which essentially defines Fe prime as the most available iron species and the iron DFB or siderophore bound iron as the least available iron species. And you can see that basically the values plot nearer to the Fe line prime, implying that dissolved iron availability in the ocean is much more available than siderophore iron. We can now take this single cell K-in and divide it by surface area and average it per station. And these stations are plotted against latitude. And you can see here some scatter in the bioavailability values of iron in different ocean regions or stations. However, these differences are not that large. It's about 5-fold between the highest availability to the lowest availability, and therefore we averaged all these values and produced the blue line here and here, which is basically a grand mean or an average in situ availability of iron. We can now compare it to previous studies and specifically look at this yellow and red line, which is our recent study with natural seawater but lab cultures. And we can see a very good match between water exposed to sunlight in an experiment and in situ ion availability. So we conclude that indeed in the ocean and the cells that we analyze, which are from the surface, photochemistry is an important component increasing iron availability. To extend the applicability of our proxy, we also normalized it to carbon. We found that carbon and surface area normalized proxies are exchangeable. The data here is replotted against water temperature, and as before, we see some scatter, but not a huge scatter, allowing us to extract average in situ availability for iron in blue, which is comparable to other studies. Overall, we can see weak to no effect of temperature, dissolved iron concentration, and phytoplankton taxa on availability. We can now move to application of our constant, and the first one is use it to calculate uptake rate in the ocean. So basically, we can take the constant multiplied by measured POC or dissolved iron concentration and obtain uptake rate. And to demonstrate that it really works, we are presenting our calculation for different studies. Our calculation are H values, and the values obtained, uptake rate measured with radioisotopes in different studies, are presented in color, and as a whole you see quite a good match, at least for some of these studies. We can also use our constant to calculate the biologically driven residence time of iron in the ocean, which is typically done by calculating the inventory divided by uptake rate. Plugging in our constant, we actually obtain a calculation that needs only POC concentration. So if we now take the common POC concentration in iron limited areas of the ocean, they range between 2 to 10 micromolar, we can evaluate iron residence time in this region to range from 2 to 12 days, which is in agreement with published values. The last application that I show today has to do with how we constrain the prediction of Earth models with our constant. I present here a study by Taglibor et al. Showing a change in primary productivity, net primary productivity, in response to climate change. And there is a very large range of predictions from their models, either a strong drop in primary productivity or actually an increase in primary productivity. And the reason the models vary so much has to do with the way they parameterize biological iron uptake. And here our constant comes into play, and we can use it to constrain their Fe uptake scenarios. So basically what you see here is one of the cruises that we analyzed. It is the Equatorial Pacific Biocomplexity cruise from 2004. And we have data from all these stations, single cell quota data, iron concentration, and therefore we are able to calculate Ke normalized per carbon. And what Al basically did, he took his model and calculated from the uptake rates in the model under high uptake scenario and under low uptake scenario, he calculated the Ke uptake constant. And we now compare what he calculated from the model from the values we extracted from the actual data from the cruise, and you can clearly see that there is a better match between our data and the low uptake scenario of the. Going back to their data, we can now indeed constrain the different options. The options we support with our data is the red line, and basically it indicates that the predicted change in net primary productivity in the Pacific in the future due to climate change is actually be very moderate, or there is even an increase in primary productivity rather than a strong decline. So it is time to conclude. Unfortunately, I kind of ran out of time. So I will show you all these amazing conclusions, I think, and we'll let you pause the video and actually go through it slowly. But I do want to take the time to thank you all for listening and thank the many, many people who contributed to this study, specifically Robert Anderson, who really pushed us to go to this very, very long trip, and all the other people for giving advice, data, and sharing their insights. And clearly, we thank the funding. Thank you all.